there was a single event where a eubacterium was incorporated into a larger cell, which gave rise to the mitochondria of eukaryotes and eukaryotes in general. This symbiont's DNA, much of it was actually then incorporated into the eukaryotic nucleus. And this can occur a number of ways. So, for example, if the symbiont is to lyse and the little cell dies, well, then its DNA is released, and this can be taken up by the nucleus. Also, there is a process known as retrotransposition in which RNA, which normally is transcribed from DNA, can then be reinserted into a DNA chromosome, and the next time the chromosome replicates, it then becomes another DNA copy. The original symbiont, which was a free-living bacteria, was estimated to have possessed about 3,000 to 4,000 genes, and about 800 genes in the genomes of eukaryotic nuclei, including those of humans, seem to have resulted from this bacteria. So when you compare sequences, they seem to have originated from a proteobacteria and thus have a different ancestry than other eukaryotic genes. Many to most of these genes do not produce proteins which function specifically in mitochondria, but rather have other roles throughout the cell, interestingly. Most of the proteins which function in mitochondria have thus been encoded by nuclear genes, some of which may have originated in this endosymbiont. And the proteins which were produced in the cytoplasm are imported from the cytoplasm into the mitochondria. Several hundred proteins function in the mitochondria of protists, while several thousand proteins function in the mitochondria of vertebrates. So these are produced throughout the cytoplasm of the cell for the most part. The mitochondrial chromosomes found in eukaryotes today are rather small, typically only encoding several dozen genes, particularly those for the proteins of the electron transport chain used in respiration, and for the translation of proteins which occurs in mitochondria using the tRNA and rRNA genes encoded by the mitochondrial chromosome. There are protists, which have the largest mitochondrial chromosomes, which can encode 66 proteins. This is the largest number. Meanwhile, there are other protists which have much smaller mitochondrial chromosomes, which can encode as few as 2 or 3 proteins. Mitochondria in diverse eukaryotes can vary. So, for example, there are protists which possess mitochondrial genes that other eukaryotes lack, but these genes are homologous to those found in bacteria. The number of base pairs can vary. So while the mitochondrial chromosome of humans has, say, 16,000 base pairs, that of a plant can have more than 2 million. The number of genes can vary, with protist and plant mitochondria typically having more genes than those of mammals. The gene order of mammalian mitochondrial chromosomes is constant, so all mammals have the same genes in the same order. The percent of the chromosome which is coding as opposed to nucleotides which doesn't code for RNA or a protein can vary, as can the number of chromosomes per mitochondrion. In vertebrates it is typically 5 to 10, but in plants it can be 20 to 40. What do mitochondria do? Well, interestingly, that varies. We are familiar with the mitochondria which perform aerobic respiration. However, there are also anaerobic mitochondria which function without oxygen and simply produce fewer molecules of ATP per glucose molecule in some invertebrates. And then there are derivatives of the ancestral protomitochondrion which no longer have chromosomes, and we no longer recognize them as mitochondria, but rather as hydrogenosomes. They produce hydrogen gas as glucose is fermented, and they are found in some protists and some fungi. There are also very small structures known as mitosomes, which do not use oxygen and produce clusters of iron and sulfur atoms, known as iron and sulfur clusters, that are found in some protists, including some of the most primitive protists like Giardia. In fact, while the ancestor of mitochondria was probably capable of using oxygen, it might have been the production of hydrogen gas, which was originally what benefited the host cells. So the archaea, which are related to eukaryotes, they use hydrogen gas. And so it may have been the hydrogen gas production, which was the original purpose of the endosymbiosis. This would have occurred at a time in Earth's history when oxygen levels were lower than they are today. In humans alone, mitochondria are capable of so many other things. The Krebs cycle is not only for energy production, but also it is involved in metabolic pathways for many other required molecules. Mitochondria can affect the transcription levels of genes in the nucleus and help regulate them, including for the cell cycle. Mitochondria can produce reactive oxygen species, which are involved in the differentiation of cells and program cell death, which helps prevent cancer. And so if the derivatives of once more complex cells are capable of all these functions just in humans, imagine how many different functions that mitochondria might have in different lineages of eukaryotes.